Welcome back, everyone. The breakout rooms are now closed, and we are all here for plenary session number 2. In this session, we have one distinguished speaker. To introduce the speaker and chair this session, please welcome once again Professor Dr. Jennifer N. Harikrishna.
Thank you, Camera. Good afternoon, everyone. It's my pleasure to introduce to you our plenary speaker, Professor Dr. Pat Hislop Harrison. Prof. Pat is Professor of Plant Cell Biology and Molecular Cytogenetics at the University of Leicester in UK, and also Principal Investigator of the Plant Genome Evolution Group, South China Botanical Garden, Chinese Academy of Sciences in Guangzhou, China. Prof. Pat was also an academic icon of the University of Malaya, and he continues to collaborate with researchers at the Center for Research in Biotechnology for Agriculture, CEBA, at UM. In Leicester, Pat, together with Dr. Trude Schwarzacker, leads a research group investigating the genomics and evolution of plants and animals with a focus on crops. As well as his prolific scientific publication, Pat is involved with media discussions and presentations, giving research advice and formulating policy, and regularly presents talks at research conferences. And for those who look, you will find he's very active on social media. Until recently, Prof. Pat was chief editor of Annals of Botany, where he still serves on the editorial board, as well as holding editorial roles for the journals Theoretical and Applied Genetics and Plant Reproduction. He's a member of various UK and international grant and institutional review bodies, and was a member of the management committee of the global Musaceae Genomics Consortium, which later merged into Musaceae. I could go on with his very prolific CV, but I'm sure you'd much rather listen to Pat speak. So I now invite Pat to give his plenary presentation, The Banana Pangenome, Evolution and Exploiting Biodiversity.
Okay, sorry, I've forgotten the mute button and all that. Thank you very much for the generous invitation, and I'm so sorry that I'm not able to join with you. I was really excited when the invitation first came, thinking, oh my, this will be a great opportunity to be in Malaysia again. Sadly, here we are a year later, still all sitting at home. Anyway, I'm very happy to have the chance to tell you today about our work with banana and putting it into the broader and I hope thoughtful context of how we can exploit biodiversity. Now, of course, from the talks that we've heard already, our knowledge of this subject, we're all very familiar with banana, both as a crop for commercial plantations, for one of the most popular fruits, and also very important for the smallholders throughout the world. I'm just trying to put my spotlight on here. Yeah, okay, good. So I'm going to be telling you about some of the work on biodiversity, but particularly I want to be more thoughtful so we can think about some of the different ways that banana can be used. And here's an example of commercial crops on the left, and smallholder systems where the banana is grown underneath date palm in this case, and above various herbaceous species like yams and legumes underneath. We're all very familiar with banana having a pan-tropical distribution now, but of course, as been said, the origin is in Southeast Asia, including Malaysia, and that's a big area. Now, a major point of my talk is only going to be talking about within banana variation, but also the importance of the whole of the group. So moving across to the sister genus of Ensete, and we plotted those distributions in there. But all of these are basically warm and humid tropical areas that they grow in. The production is, of course, in the tropics, and I like this distorted map here showing India as by far the biggest producer in the world, but of course little known outside India since there's minimal export of banana from there. South America, the major export into Europe, particularly in the US, and then of course a very significant production in Malaysia, Southeast Asia, where there's both export and local production. Just to show a picture of variation in banana, it's in an Indian banana shop here, and you can see the huge range of different varieties. And even in Malaysia, there's only 5 or 6 that are regularly available in the shop for a number of different problems that I'm going to be talking about in 3 slides further on. So what does the taxonomy look like in this area? Well, as it's shown on the right-hand side, Darwin 170 odd years ago had it right that there was a common ancestor, a most recent common ancestor, diverging into what we see currently in the right-hand side in its origin of species here. And here's a more modern species of the Musaceae, the banana family, with the bananas and Musa acuminata and balbisiana, the wonderful names of our breakout rooms. Then many other species like Textilis, itinerans, and so forth. And then sister to that, there's the genus Musella. Sister to this group, Musella, the sister is the Ensetes, which are important, so Ensete ventricosum particularly, is important starch crop. But because I'm talking about pan-genome, I want to ask the question as to how much this variation is available for breeding in banana. Just so you see what Ensete looks like, it's not eaten as a fruit. In fact, the plant is cut down short as soon as it starts to flower, and the base of the pseudostem and the corm and leaf bases are very highly productive starch crops. And it's a staple food in Ethiopia in particular, with a very high production of starch. So the starch is stored in the plant. It would then be mobilized and put into the fruits, but actually the plant is cut, and then this is fermented and boiled to produce starch. But both Ensete glaucum, Ensete ventricosum, and Ensete glaucum that I'll be talking with have very interesting extra genes, which we think are certainly going to be valuable for addressing some of the challenges in banana. And of course, we also go beyond banana into the Zingiberales, ginger order, and with Jenny is involved in sequencing one of them, finger root ginger, Bosenbergia, and that is one of the sources of genes for the whole of the Zingiberales, which can certainly be used and transferred into banana using a range of different technologies. And of course, we also need to think, what about all plants, and how much can the whole gene pool of all plants over the last 450 million years since the plants moved onto the land, how many of those genes are available? So the work that we and many others are doing is really trying to put the whole of the plant genome evolution into a bigger context, and then how can we use that particularly within the banana group as in here within the monocots. So banana, of course, has a lot of challenges, and we've been hearing about those. And this is a rather wordy slide, I'm afraid, but I decided that I wouldn't split it up into many different slides, but put it all in one case here of the challenges for banana. And of course, I would ask anybody among the farmers and growers, what are the most pressing needs that you have? Of course, from the consumer point of view, we are wanting cheap price, high quality, storable, and so forth. But farmers' challenges can be very different. It's very important for geneticists like myself to be in contact with the farmers as well as the breeders so that we can help to address these different challenges. I always talk about the F's of farming here, and of course, banana particularly provides us with food and fiber. Other crops provide us with feed for animals and fuel to burn or to ferment, and then other things, flavors, horticultural industry with the flowers, the fun of native environments, or where we plant plants for enjoyment, whether that's in golf courses or in woodlands and so forth. And of course, the final F of pharmaceuticals, where plants provide us with a lot of the pharmaceutical origin components, and we need to think about those. So all these F's of farming are very important for the outputs of what the farmers are producing. I want to show on the right-hand side then some of the big challenges that we have. And of course, in the breakout rooms this morning, we've been talking about abiotic stress, when that's water, temperature, wind, climate change was mentioned in one of the sessions. And then, of course, the critical biotic stresses. Then plants, just like humans, suffer from fungal, bacterial, and viral diseases. And we really don't want to use drugs and agrochemicals where that can be avoided. So how can we change and reduce the use of those from the genetics in order to give resistances and high production even under those? Of course, that also links into agronomy. We can do a lot with agronomy to overcome some of these stresses, but also for banana, can we expand the area of this highly productive crop, not to replace wild areas, but to replace other less productive agricultural products? Post-harvest is incredibly important, and increasingly so. Still in Asia and Africa, there's increasing urbanization of the population. The world's population became 50% urban about 25 years ago, and in the last couple of years, the African population has become largely urban. Of course, that means that all the fruits, and particularly bananas, have to be harvested, sold to middlemen, transported into the towns. And these are all giving challenges for transportability and storage. And of course, if you ever visit a tropical market, you will often see behind, in the back of the stalls, huge amounts of bananas being disposed of. And that's after all of the water, all of the agrochemicals, all the nutrition, all the care of the farmers. It's never made it to the person that will eat it, because it's not been possible to transport, store, and sell it in time. So we really need to think about those to reduce the impact of the agricultural that we're carrying. I've talked about this, socioeconomic uses, but we also need to think then very much about what we can do to increase the market and increase the profitability for farmers of banana. And that certainly includes new uses, whether that's through processing banana chips, and then now ultra processing is the really big market. We're seeing in the UK, plant meats are now available absolutely everywhere, being sold for more than meats, but ultra processed foods are certainly a very big trend in the marketplace now. There are other products, waxes, fibers, starches. Starch I've already mentioned as a key product from the Ensete, also from banana. We can use all of these different things. Of course, the Musaceae family, banana, the fibers are used, first of all, the leaves being used as plates, that I'll illustrate in a couple of slides, but also the Musaceae textilis, abaca, or manila fiber is a very good high quality fiber and we should really be looking at where we can sell banana products, Musa products to the big companies to improve the sustainability, recyclability. So why is a car dashboard made from plastic? Why isn't it reinforced with banana fibers to make the quality there? And these, of course, are very high quality, high value markets. Medical uses and medicinal uses. The bananas Ensete have a number of current properties which are claimed. We need to get proper evidence for those. And we need to also think about new properties. Of course, the vaccines and antibiotics have both been around and suggested. And maybe now there's been wide acceptance that people can be GMs. So I expect I and most of the people in the audience may are by now GM with the adenovirus or mRNA vaccines. It's just crazy that we've not been able to do that during the whole of my career, basically in the plant world. So maybe legislation will be changing. This was mentioned during the introduction. Maybe legislation will change to allow that. So I want to just illustrate a few of those points, but just think in the back of your minds in the audience, what are the diversity and which banana varieties have you seen that have better resistances or better qualities or better or different uses that you've been able to buy in the market. The germplasm pool is where most of this variation is coming from, though. And here's an example in Malaysia in the left-hand pictures. Here you can see Big Mike Gros Michel banana, totally taken out by the Fusarium, Panama disease, tropical race 1, not tropical race 4 that we're hearing about. But here it is lying dead in a field in Malaysia, whereas next to it, there's quite a productive plant here that's resistant to that. You can also see in this field another sustainability problem, and that is the runoff, the water running off all of the soil in this field here, and that of course is a big problem. And I always ask in some of my lectures, how far is humanity from extinction? And people come up and say a few million years, or oh, they'll find solutions or whatever. My answer is about 15 centimeters, the average thickness of the topsoil. And if that is washed away, then we lose production. And that, of course, is an important part of sustainability. But equally, it's an important part of agronomy. If you do have too much, if you have grass around the banana plants, then the allele chemicals will be fighting against the bananas, and therefore the production will go down. So maybe we need to consider the whole of the production, the whole of the sustainability of the field production. And then on the right in China, we can see the tropical race 4 effects on the banana. Of course, about 30% of the production cost of banana is in the agrochemicals and the crop protection costs, whether that's all these different things that are sprayed to control diseases. And then, of course, it's a very important part of agronomy. And here we can see plants affected by banana bunchy top virus being rogued and burnt very sensibly by the farmers in this village that carefully rogue every plant that's infected. The other uses I talked about, here are some of the different processing uses which people will be familiar with, and of course the banana leaf with the banana curries and so forth here as a starch. And of course, agronomy and having clean planting material is very, very important. And tissue culture has revolutionized, led in many cases by the technology in Malaysia, has led that revolution so that virtually all the plants are now grown through tissue culture. That means that the plants go into the field without disease, biosecurity is very, very important. And here, for example, are the sort of information from one of the strongest countries in biosecurity, whether that's human diseases like COVID or plant diseases. Maybe they're basing their human rules on what they've thought about, what they know about plant biosecurity. And here's a sort of very strong message that the Australians give about the introduction of banana freckle. Don't do them, destroy them or remove them yourself, but get people in who know about that. Do not move bananas. So this is all a way that we can control the disease spread, and that's sort of part of the agronomy in the way that they're being grown. So I talked then about identifying the challenge. We need to identify useful genetic variants in order to bring that. And one of the things that Biodiversity, an international consultative group on international agricultural research unit, Biodiversity, now Alliance of Biodiversity and Biodiversity and SEAT, is to identify and collect useful genetic variants and make those available for the breeders. And this was a document which about 50 people put together about the conservation strategy for conservation and use of Musa genetic resources. And this has been this is a very important group, a very important aim to conserve and make sure that we're not losing those. Because in a way, the people, geneticists like myself, we're slightly schizophrenic. In one hand, we want the farmers to have available to them and to grow the improved, highly productive, low impact, sustainable varieties, which are the best for their areas. On the other hand, we don't want to lose what is out there of genetic resources. And there's challenges there of both which varieties farmers are growing and also loss of habitat where they might be growing in the wild. Another very important part, though, about conservation and use, which I often find myself talking about because my job is at the universities, and we spend a lot of time talking to national institutes like Embrapa in Brazil or Biodiversity, Favre in Vietnam, and so forth, but is the importance of training and tertiary education. So that is a very important aspect of making the best use of what genetic resources that we have and knowing how we can reduce the impact of agriculture on the environment. Germplasm resources, of course, are grown out in the field. This is one in India. I've already shown some from Malaysia. And there's a fantastic Biodiversity and Crop Diversity Trust website with the Musa Germplasm Information Service giving the data of some 6,000 accessions of banana. So it really is a remarkable amount of diversity, even although, as Jim Lawrence, and I'm afraid I wasn't up at 2 o'clock this morning to hear his talk, but reading his abstract, he says just that more than half of the pool of more than half of the bananas eaten in the world come from just 2 small pools within these 6,000 accessions. So it really is there really is quite a bit of variation within Musa, within banana itself, but actually there's even more, much more broadly. I showed an old picture here from Theo, now at University of Malaya, but it's so nice showing the variation between different bananas by the fingerprint much more graphically than a few A's and T's in a modern picture. But then that's showing here how we can build up a phylogenetic tree. I showed in an early slide the complete phylogenetic tree of all plants. Now we're just looking at the bottom of that tree, just at the separation of Musa valvisiana and Musa acuminata. based on DNA genetic markers. If we look at the whole of the tree here, this is a whole range of different bananas, and you can see the Cavendish, Williams, Granane, Pangenome are all in a very small group here. But there's a lot more variation around, both with the A genome and the B genome. Now it would be much less graphical, and this is a more recent analysis. In this case, the diversity of Ensete using genotyping by sequencing. So instead of a few hundred tracks on a gel, the last picture was constructed, a few tens of tracks. Now we've got 600 megabytes of sequence data from hundreds of accessions, and we can cluster those. The blue ones are the wild species, and the red and the green are showing the clustering of domesticated ones that are grown in here in Ensete. So that's this sister group here. Now let's look in here. So what's the first big difference when we get down to the domestication? Well, of course, there are very critical domestication traits, and this can be one of the difficulties that we have in bringing in genes from wild species, that we also bring in the bad characters that we see like these. Of course, seedlessness is one very important one, and that's not just because the seed doesn't set. It's what we actually have when we eat a banana. It's like having in human terms, like a placenta without an embryo, and normally a fruit without seed will abort. So there's very complex relationships which really aren't very well understood yet about how the banana fruits are developing without the presence of the seeds. This is a character which is very interesting. This is one that's Musa velutina, and that's the self-peeling in extreme, where as the fruit ripens, the peel just comes back like that. And that's one of the reasons that Cavendish is so desirable in the marketplace, because it doesn't peel at all like this. Whereas if many of you, I'm sure, have brought variety like pisang awak and so on back home and discovered that half of the fruit has fallen off the bunch in the supermarket trolley. So that's not a good character, and it's quite complex to have that removed, and is one of the challenges even within the banana germplasm pool, let alone going more broadly into other Musa and Ensete. I again want to show another little bit of older work that we did with Azhar Mohammed at Nuclear Malaysia, MINT, Malaysia Institute of Nuclear Technology, looking particularly at disease resistance genes across banana family, across the whole or all species of banana. And here we have this nucleotide binding domain and the leucine-rich repeat, which all help cross the membrane and help to give the banana resistance to many different diseases. This is the parallel lines here indicate a repeat of a leucine-rich domain. So you can see with the protein sequence at the top there. And that led us to looking at a range of the genes with these nucleotide binding sites, leucine-rich repeat genes, from the Musaceae textilis, velutina, the self-peeling one, and ornata. And altogether, we found quite a lot of interesting variation here, which can certainly be brought together. And here we can see some screening, including a mutant variety, which is another way to alter control of disease resistance, and the Mutiara variety, which is part of the Malaysian Pistan-Rasali complex, which is giving good tolerance to both race 1 and race 4 of the fusarium. So these are some of the type of ways that we can look at variation and find it. Another piece of work I want to talk about was the banana streak virus, which gives these symptoms on leaves and eventually no disease, eventually no yield. a small DNA double-stranded virus, about a 7,000 base pair genome, which is integrated into the genome. It was actually the first virus that was found to be within the chromosomes in the blue picture on the left of plants, but now quite a widely known phenomenon. We could see how it was organized in the nucleus. And 20 years ago, we showed that with in situ hybridization. And then more recently, we're involved with the banana genome sequencing. And here you can see the complex structures of that linear banana street virus being incorporated into the nuclear genome, just as we had seen by looking at the DNA fibers in the nucleus. Now, what is this doing? Well, one of the things might be this is the plant using the mechanism which we're now using to give resistance of RNA interference. And it's most likely that these pieces of DNA in the nucleus are being expressed. Those provide primers, which then lead to the destruction of exogenous banana street virus. So the plant is gaining some resistance to the disease by the internal genetics of the genes, the incorporation within the DNA of these banana street virus pieces. So I now want to give some of the new results that we have with the Ensete group, which is a sister and has a lot of common genes which are available, as I'll be showing, between ventricosum and glaucum, Ensete ventricosum and glaucum. This is some work with Manoj Biswas here, who's shown how markers, in this case simple sequence repeat or microsatellite markers, are shared between all of the Musas and Ensete in here. So we have a relatively large proportion of markers which are transferable, showing that there's a common gene pool there, even more within the branch leading to the Ensete ventricosum. More than half are there. We used those markers for one study with Gizachu in Ethiopia. And here we were interested, he was interested in the medicinal banana Ensete that has medicinal uses. Our null hypothesis was that the domesticated ones going for starch would be less variable and a particular group of bananas but Ensetes, whereas the medicinal ones would be much more diverse because they've been selected for different properties. But our null hypothesis was very clearly wrong. And you can see here the variation of the non-medicinal blue varieties here is really within the cloud of variation within the medicinal ones in orange color. And here you can see the whole range of uses that the Ensete is being put to, and the green ones are the starch varieties, and you can see those are all over this tree showing the relationships of the different species. So it's not that the domestication has selected a relatively small pool of variation in there. We then went on to look at how variable genes are between all of these species in the terms of what gene families are present. So here we have Musa balbisiana, Musa acuminata, top and bottom, and then Ensete, and another banana here. And you can see that of the 36,000 genes that are totally present there, we can say that about 2/3 of those, just under 2/3 of those, are common between virtually all of the species. So that's the nature of the gene, not the detailed sequence. I'll talk a little bit about the detailed sequence in a few minutes. So there's an awful lot of genes that are shared. There are relatively few that are specific to the Musa branch, 600 out of 35,000, and 1,500 or so that are specific to the Ensete branch. So that's interesting, indicating that there are some new genes which might be useful to be used in banana if we want to change some of the properties. And the Ensete glaucum is particularly interesting because it's the most resistant of bananas to abiotic stress. It's the only banana that will grow in snow and at higher latitudes. So can some of that biotic stress, which may also be related to abiotic stress, be transferred? We're just working on this paper led from Dai Wang and Qinglo at South China Botanical Garden, Guangzhou, and here we can see exactly what the range is. Had a lot of it's all in collaboration with Gert and Drock and Matthew Ruad and Nicholas Roux, Frank Christoph Barons at CIRAD, who put together the genome browser and done a lot of the analysis here. So what happens then when we look at gene families between these? Well, if we look, we can see here's the most recent common ancestor of all of these monocots here from the Phoenix. And I don't know why my screen seems to change the sizes of the images here so that they don't match up. But we can see what the same as we saw on the other considerable number of gene families which are expanding in the Ensete group, and of course a very large number that expand in the Musaceae in here. So let's look at an individual group of genes here, and those are the starch synthase, which is so important both for the fruit and the corm in the starch group. Here's the number of genes present in an analysis that Manoj Biswas has done for the paper. Starch branching enzymes, starch synthase, and other branching enzyme, and AGP, glucose polymerase. And here we can see that there's actually rather similar number of genes in all of those. But our provisional analysis shows that one of the granule-bound starch synthase, you can see that down this is one member of the family, and you can see that it's present in Ensete glaucum, Musa balthusiana, Schizocarpa itinerans, and acuminata, acuminata, similarly here, present in most of them. But interestingly, the provisional analysis showed that there was a triplication of this particular branch in the Ensete glaucum, and that's giving rise to factors to the wider and more starch synthase genes here. So some of this indicates where gene expansion might relate to function. And that's different from the polymorphisms, which of course we're also very interested in looking at. Another key thing is how do we use this variation? Well, one of the problems is that chromosome numbers differ, whereas the Musa acuminata and balthusiana have 11 pairs of chromosomes. The glaucum has only 9 pairs, but it's very interesting to see how actually there have been various fusions and fissions of the chromosomes relative to each other. So Ensete glaucum has most of chromosome 9 of Musa acuminata and half of Musa chromosome 7. The chromosome 9 of Ensete glaucum is entirely represented in Musa 11. One arm becomes a single chromosome of Musa acuminata of banana 11, and the other arm becomes a large part of banana 4. So this gives some optimism that we can actually transfer these genes potentially by crossing. There'll be a lot of work, but it does mean that we can look for rearrangements which we might be able to do as breeders. That's showing the blocks of how the genomes have changed. Two genome sizes are about the same, so most of these lines are relatively similar there. I want to point out the importance of the Banana Genome Hub from Gertin Drock, Matthew Ruard, and colleagues here. We weren't involved with this, but it includes our data now. It shows the whole DNA sequence of all these species. It has underneath that the RNA and expression data, and you can go in and look at exact variation of all of the genes that have been found across the banana genome. And it's a fantastic, publicly available, fast and available resource. You can see the sort of thing here, for example, in this is actually the same GBSS granule bound starch synthase I showed before. You can see here there's 2 different amino acids. It's variation shown in that in the particular sites underneath where my pointer is. So it's very important and extremely valuable. And now it's very important that academic researchers populate this even more with more variation data and more sequence data, so that we can link that with what the breeders and geneticists, field geneticists are finding of genetic variation, and then using that. So how good have people been in improvement of crops? Well, this shows 59 years, 59 years from 1961 to 2019 of the yield of major crops compared with the number of people, which is the red, maroon, dark red line at the bottom. And you can see it's a very positive story. People have grown more of rice, maize, and wheat, major crops, over continuously increasing and a steeper slope than the number of people increasing in that 59 year period. But what has caused this? Well, I've decided this is 3 different phases. Agronomy in the 1970s and 80s, when people realized that you could fertilize things, you could control disease much better, have the spacing of the plants and so forth. And that was all the first stage. Then we move on to genetics, where crop genetics was improved. And finally, we got to the new breeding technologies, which have been particularly led to the increasing in the slope for maize, where the disease resistance, insect resistance, and weed control have improved through the GM methods, and that's given that an extra boost in that crop. Unfortunately, the picture is nowhere near as positive for banana, and in the last 10 years, we've seen a very static yield of the crop, and we've completely broken the previous 50 years of regular increases in banana yield globally. The Malaysian statistics are very, very bumpy. I don't really know why, but they keep changing the way that the banana yield is being calculated, or the banana production is being calculated. So it's less informative than the global picture here. But really, we need to do something to stop this marginal decline in yield and improve the quality. So the future of banana crops, gene power has diversity which can meet the challenges that I talked about. Breeders need to get better, and banana has these major extra challenges for the breeders, including post-harvest things like the transport and ripening and the sterility and triploid nature. So we can use the diversity, crossing varieties, genome manipulations. That's certainly how I think the genes one way genes can be used across the family, chromosome manipulation, generating recombinant, and of course, a very important gene editing and transgenic approaches, which hopefully we'll be able to introduce now to plants as well as to people. So communication, collaboration is very important. What we really want to do is to have sustainable production, thinking about the circle here, inputs being converted into our outputs of crops, but also the features of the land, modulating water, gases, nutrients, carbon dioxide outputs from the way that farmers are growing it in the cycle around here. So finally, I'm very proud a decade or so ago to get a letter, actually joint letter in the Financial Times to the economists, saying that we wanted to draw attention to the discovery that technical change was the biggest source of growth, and that seems to have been forgotten. So scientific advances are really going to help in this, as in all other aspects. So I hope then in the last, I'm afraid I've overrun a little bit here. Banana production faces lots of the challenges. We need new genetics. We need to identify the challenges, identify useful variants and use those. And I've shown some of the modern ways that we can measure diversity. And we do need the field work as well to how we can use this common gene pool to improve banana for sustainability and for the farmers. I will put my slides onto SlideShare after this, so they're available, but very happy to talk, answer any questions, and please contact me if I don't reply right again, because I do get buried under the email and not particularly good at replying. So thank you very much.
Thank you very much, Pat, for that very comprehensive tour through, I think, the bananas and important relatives as well. So that was, I think you've really shown how looking at the diversity of genetics in the Ensete has allowed you to, show possibilities for new genetic materials within the relatives, and I think that is something that we've heard today is going to be very important, especially for those of us in this region where we talked about our Banana Hunter project. Really, we need to look at our own materials and the resources that are here. I don't see any questions in the box yet. Does anybody have any questions for Pat? I think I will start with one though. If we are planning that we should really look at all the germplasm that we have, especially in this region, and I think we have colleagues here from Indonesia and Philippines and other countries where banana is native, China, Australia. So what would be maybe the main thing we should be looking for? Do you think it's disease, or is it climate change, or is it new products? Where should we be putting our energy?
Well, can I just go back to that slide? I was, as much as anything, it was really a question for you, for the people in this audience who are involved, the farmers, growers, producers, and so forth. We now need to study the whole genome. The days of just looking for one particular gene at the academic level, we're past that, I think. The breeders and maybe the national programs could certainly be doing that. But we need to assess the whole range of variation, which is present there. And the new genomics methods let us do that. And then we can link those with whatever characters the field geneticists, the breeders, and the farmers have identified in their material. So I think we need to take at the academic level a more holistic approach, but the people on the ground need to think about specifically which genes and what improvements they're needing to address. And that's partly what I mean by super domestication in one of my last slides. How can we need to design and think about what we need, and then it can go, then the ways to find those genes or engineer those genes, transfer those genes, can be passed to the genetic engineers, genetic technologists, who can decide the most appropriate ways to introduce and select the genes to meet those challenges.
And another big challenge, I think it has been alluded to also, I think by one of our items this morning, is that it needs a concerted effort. I think not just a lot of people, but also a long-term plan that it was mentioned, I think, by Dr. Gurmit, that maybe we need a banana council, just as there are equivalents for other important crops in Malaysia. But
yes, absolutely. But yeah. I hope that the Banana Council will also include training as part of its remit, and particularly in training the next generation of senior scientists and the generation of people who will work with the farmers.
Yes.
I'm always impressed by India, that has tens of millions of people trained to MSc level, who are out working with the farmers and showing how the plant, how to grow the new varieties, how to check the diseases, ensuring that, for example, the banana bunchy top plants are burnt immediately, that fusarium areas are no longer put down to banana, whereas in other cases, new varieties might redeem the yield penalty from an introduction of a disease. So training is needed, and of course that's very, very long term as well. That's a generational issue.
Yes, well, I think that also goes to our loss of pure botanists and taxonomists as well. That that is maybe a too big a topic for today. But I think in interest of time, and since I don't see any messages, and I think people also know how to contact you, Pat, if they want to, or you can leave messages in the box. We can forward those to Pat. I think I will have to close this session because we're actually quite late. And,
sorry about that. Yes.
Also, you started late, so I didn't want to stop you. And thank you very much for that really interesting presentation. I think it's given us a lot to think about. In fact, the whole day we've had a lot to think about. So we all need to be ready to start again tomorrow. But before that, we have one more exciting session. So I'm going to pass it back to the MC now to take over. Thank you.